Hello. Let us today discuss about the unsung hero, the superhero of balance between gluconeogenesis and glycolysis and gluconeogenesis. Okay. So this PFK2, also known as fructose 2,6 bisphosphatase, this is the enzyme which is the unsung hero. Why do we call it? Because one, it is bifunctional. Two, it is balances both glycolysis and gluconeogenesis. The question is how does it do it? Okay, so as we are thorough with the glycolysis and gluconeogenesis. These are the small simple steps which we'll discuss now very quickly. Okay, so here coming to this part, in this glycolysis, the glucose is converted into G6P and F6P, and from there it is converted by PFK1 into fructose 1,6-BP, and again at last the pyruvate, and then to acetyl-CoA by PDH complex. So there are few common elements which are common for both glycolysis and gluconeogenesis. They are glucose, that is common, G6P is common, okay, fructose 6-phosphate is common, fructose 1,6-bisphosphate is common, okay, most of them, okay, and PEP. These are the few important things. This PepCK is an enzyme, okay, phosphoenol pyruvate carboxykinase, PepCK, okay. So what happens here? When pyruvate produces acetyl-CoA, this acetyl-CoA inhibits the PDH complex and then promotes the pyruvate carboxylase enzyme, which causes the synthesis of oxaloacetate from pyruvate, which goes into the gluconeogenesis pathway. This is during the fasting state. So what is favoring this this pathway to occur? See, when this fructose 6-phosphate, it is the it is like the underworld agent, okay. This is underworld agent. It indirectly controls. Why do you call it underworld? Because this phosphofructokinase enzyme, this phosphofructokinase 2 enzyme, this synthesizes fructose 6-phosphate into fructose 2,6 bisphosphate, okay. This is good as far as this is concerned. Up to fructose 2,6 bisphosphate, it is good. This phosphofructokinase, it is very important, okay. Remember this fructose 2,6 bisphosphate, what it will do is that will inhibit the fructose 1,6 bisphosphatase of the gluconeogenesis cycle and promote phosphofructokinase 1 of the glycolysis, okay. As far as now, are we clear? Good. So this fructose 2,6 bisphosphate regulates these two enzymes of glycolysis and gluconeogenesis, but how to stop it? Okay, this phosphofructokinase 2, see, this is what happens in normal fed state. When you take food, this is what happens, okay. And we will now discuss about the superhero of today, that is PFK2, okay. So what happens here? This fructose 6-phosphate is converted into fructose 2,6 bisphosphate, which in turn inhibits the fructose 1,6 bisphosphatase, okay, and promotes the phosphofructokinase 1, okay. This favors the fructose 6 phosphate to enter into glycolysis, okay, and this favors the fructose 1,6 bisphosphate to enter into gluconeogenesis. Here also glycolysis, fructose 1,6 BP is formed, bisphosphate is formed, okay. As far as this is the step we are concerned about. So what is with this PFK2? Okay, this fructose 6 phosphate itself, this fructose 6 phosphate itself controls the PFK2 enzyme. The availability of fructose 6 phosphate controls the PFK2 enzyme. So what is with this PFK2? What happens when PFK2 is there? It has two moieties, like two functions, bifunctional, right? One is kinase activity and other is phosphatase activity, okay. What is this? This kinase activity makes the PFK2 act as PFK2, isn't it? The kinase. This phosphatase activity makes it to act as, can you tell? Makes it to act as fructose 2,6 bisphosphatase enzyme. So what happens is this kinase activity, this in the fed state, okay, this in the fed state acts as PFK2, activates PFK1, okay, promotes glycolysis, okay. How promotes? How does it promote glycolysis? By producing fructose 2,6 bisphosphate from where? Fructose 6 phosphate, okay, via, you understood? Good. So what happens in this phosphatase activity? In fasting, during fasting there is increase in cyclic AMP, which causes the induction of phosphorylation, okay, phosphatase activity, right. This cyclic AMP leads to the induction of phosphatase activity of the PFK2 enzyme. That means this PFK2 now behaves as fructose 2,6 bisphosphatase, okay. It causes cleaving, okay. This phosphatase activity breaks down fructose 2,6 bisphosphate, that is this and this part, okay. So there is no glycolysis, you understood? So there is no glycolysis and no inhibition of which enzyme? Fructose 1,6 bisphosphatase, favoring the gluconeogenesis, okay. So this is how it controls the balance and maintains the balance. So fed state, so what does it say? It says insulin dependence. Okay, and in fasting state, what does it say? Glucagon. Okay, so what did you understand by this glucagon and insulin? We can say that this PFK2 has two moieties. Okay, two activities or two spots that can be activated by different enzymes. Okay, so this glucagon activates the phosphatase activity, while this insulin activates the kinase activity. So we can just infer like that, right? Okay, so this is the concept. Thank you.